Welcome, A-level and FSC students. Today we're diving into this explainer on plant support, Turgor mechanics, Collenchyma and Sclerenchyma. Have you ever actually stopped to wonder how a towering tree, or even just a simple sunflower, manages to stand completely upright against the relentless pull of gravity, without a single bone in its body? Well, let's unpack the brilliant anatomical engineering that plants use to solve this exact problem. So here's our roadmap for today. We're going to cover 1. Turgor mechanics and hydrostatic support, 2. Collenchyma, flexible growing support, 3. Sclerenchyma, rigid mechanical strength, and finally 4. Comparing plant support tissues. All right, Section 1, Turgor mechanics and hydrostatic support. Let's look at the plant's very first line of defense against gravity. Before a plant gets all woody and tough, it essentially relies entirely on water. Enter turgor pressure. Think of a plant cell kind of like a balloon stuffed inside a cardboard box. When you fill that balloon with water, it pushes super hard against the inside of the box, right? That makes the whole structure rigid. This turgor pressure provides a vital hydrostatic skeleton for herbaceous stems. And get this exact same pressure mechanism generates enough shear force for delicate little root cells to literally push their way through solid concrete. Crazy, right? Now, for your exam marking schemes, you absolutely have to know the exact step-by-step osmotic mechanism that uses this turgor pressure to open stomatal guard cells. First, large quantities of ions and sugars are actively pumped into the guard cells. That drops the water potential inside the cell. Because of that gradient, water naturally flows in via osmosis. The guard cells swell up, becoming highly turgid, and they basically buckle outward. That buckling opens the stomatal pore for gas exchange. It's just a perfect micro-level example of turgor mechanics in action. Moving right along to Section 2, Collenchyma, flexible growing support. As these plants grow, they definitely need more than just water pressure to hold them up. And check out this cross-section because it brilliantly illustrates a major structural adaptation. Notice how the cell walls aren't the same thickness all the way around? They have these distinct, chunky corners. Actually, make a mental note right now. Unevenly thickened is the exact phrasing you want to drop in your A-level and FSC exams to secure those marks. I love a good memory trick, so to lock this tissue profile into your brain for exam day, just use this simple mnemonic, the 3 C's of Collenchyma. They are thickened at the corners, they contain cellulose, and, well, their name's Collenchyma. It's a super straightforward way to keep this tissue separate from the others we're going to look at. Structurally speaking, these guys are living cells. They actually hold on to their protoplasm when they mature. Their primary cell walls are packed with cellulose and pectin, but they completely lack lignin. Because they're squished so tightly together with hardly any intracellular spaces, they form these incredibly strong, flexible strands. You've definitely interacted with this tissue before. You know when you snap a stalk of celery and get those annoying stringy bits stuck in your teeth? Or when you feel the tough edge of a leaf? Yeah, that's Collenchyma doing its job, providing flexible support to the growing parts of the plant.
Okay, this is where biology gets super dynamic. Collenchyma actually physically responds to mechanical stress. If you have a young plant that's constantly being buffeted and shaken by strong winds, it will literally adapt by engineering its Collenchyma walls to be 40% to 100% thicker than a plant grown in perfectly still air. It essentially builds itself a tougher skeleton just to survive its environment. How cool is that?
All right, Section 3, Sclerenchyma, rigid mechanical strength. This is where we look at the heavy-duty solutions for mature plants. So, while Collenchyma is all about cellulose and pectin, Sclerenchyma brings in the heavy artillery, lignin. This is an absolutely crucial syllabus definition for you guys. Lignin gets deposited into the secondary cell walls, making the tissue incredibly hard, stiff, and highly resistant to decay. Botanists actually call it a wire-like mechanical strength because it completely prevents the plant tissues from tearing under high stress. Depending on what exactly the plant needs, we can split Sclerenchyma into 2 major categories. On one hand, we've got fibers. These are elongated, thread-like cells with tapering ends that give immense tensile strength. In fact, these are the exact same fibers we harvest from flax and jute to make tough ropes and fabrics. On the other hand, we have scleroids. These are short, stout, irregular little stone cells that provide extreme hardness. Think of the protective shell of a walnut, or that gritty, sand-like texture you feel when you're eating a pear. Now, what's really striking here is the shear scale of the cell wall. Take a look at the transverse section. That heavily lignified secondary wall is so utterly massive that it severely restricts the internal space of the cell. That central cavity is called the lumen, and in Sclerenchyma cells, it is remarkably narrow. If you look really closely at those thick walls, you'll spot these narrow little channels running right through them. We call these pits. Way back during the early development of the cell, before that secondary wall became fully thickened and lignified, these pits allowed substances to be exchanged so the cell could communicate with its neighbors.
Let's bring this right back to your lab work for a second. Think about those standard microscopy practicals where you have to mount a slide using overripe pear tissue and an iodine stain. Ask yourself, when you finally get your fine focus locked onto those dark, clumped groups of Sclereids, is there any cytoplasm inside them? Like, are these cells actually alive?
Well, spoiler alert, no, you won't see a drop of cytoplasm. Sclerenchyma cells are entirely dead at maturity. They completely lack a protoplast. The plant essentially sacrifices these living cells, hollowing them out completely, and leaves behind only their heavily fortified, lignified walls to act as permanent, solid structural reinforcements. Which brings us to Section 4, comparing plant support tissues. Let's synthesize all this into a rock-solid comparative analysis. Okay, seriously, you might want to grab a screenshot of this slide because contrasting Sclerenchyma and Collenchyma is a classic high-yield exam question. You got to be able to clearly state that Sclerenchyma consists of dead cells with uniformly thickened lignified walls and a narrow lumen, and that it serves the mature plant parts. In stark contrast, Collenchyma is made of living cells with unevenly thickened walls, made only of cellulose and pectin, no lignin, providing flexible support for young, growing shoots. Knowing these distinctions cold is going to secure you top marks, for sure. Before we finish up, I want to quickly point out that this structural engineering doesn't just stop at simple tissues. It absolutely crosses over into complex vascular tissues too. Xylem vessels and tracheids, much like Sclerenchyma, are dead at maturity and possess highly lignified secondary walls. And that isn't just to keep water from leaking out, it's to provide the immense structural support required to resist the massive suction force of transpiration pull, so the vessels literally don't collapse inward on themselves. Even the phloem has specialized phloem fibers dedicated purely to extra strengthening. It's just wild to think about, right? The next time you walk past a massive oak tree or see pictures of a towering redwood, just realize that the most massive organisms on our planet are supported largely by the ghosts of dead cells. They're using hollowed-out, lifeless tubes of lignin to completely conquer gravity. What does that teach us about the sheer brilliant efficiency of biological engineering? I'll leave you to ponder that one. Thanks for exploring this with me today, and keep up the great work studying.